pH denote what? Acidosis, alkalosis, है ना? Okay, today's topic is maintenance normal pH, acidity, alkalosis. So let's start with some terms about today's topic. What is pH? pH means negative logarithm of hydrogen ion concentration. Okay. Normal range pH is 7.35 to 7.45. Here it is false actually. Please remember this one. 7.35 to 7.45 is the normal range of pH in our body. Okay. What are acids? Acids means they donor of proton. They give rise to hydrogen ion concentration, hydrogen ion. Okay. So acid means they give hydrogen ion. Bases means they accept proton. Strong acids means they completely dissociates and give maximum number of proton. Strong acids. Weak acids means they give partial amount of hydrogen ion. They do not dissociate completely. Okay. Example strong acid like hydrochloric acid, sulfuric acid, nitric acid, etc. Weak acids like example carbonic acid, citric acid, like this. You may ask in the Viva exam, please name some strong acids, name some weak acids. Okay. You will ask in Viva exam what is pH. All of the students have to familiar with these terms. What is pH? pH means negative logarithm of hydrogen ion concentration. Okay. What is the normal range of pH in our body? Remember that one, not this one. 7.35 to 7.41. All of the students have to remember this. Okay. Acids means they donors of proton. Bases means they accept proton. pK value. What is pK value? This denote the pH where 50% of pK means the pH where 50% of hydrogen dissociates. On the other way, you can say 50% of acid will be neutralized and give rise to salt. So here you have acid is to salt ratio 1:1. Understand? Understand? Okay. pK means at which the acid is half ionized, like salt is to acid 1:1. What is alkali reserve? Here bicarbonate reserve in our body. Bicarbonate is CO3 minus. This is called bicarbonate. Normal range 24 plus minus 2 millimole per liter. That means 22 to 26. This is normal range. In our body, bicarbonate reserve is there. It is about 22 to 26 millimole per liter. Okay. What is buffers? Buffers are solution which they resist change in pH with some extent. Okay. So this term everyone should be familiar with. One or two questions may ask in the Viva exam from this table. Okay. So let's start with acid and bases. As I told earlier, acid are substances capable of donating proton, while bases are they accept proton. Acids are proton donors and bases are proton acceptors. What are weak acid and strong acids? The extent of dissociation decides whether they are strong acids or weak acid. Strong acid dissociate completely in solution, while weak acid ionize incompletely. For example, hydrochloric acid, the complete dissociates and give rise to hydrogen ion and chloride ion. Or carbonic acid H2CO3, the incomplete dissociate or partially dissociate and give rise to hydrogen ion and bicarbonate. Okay. Next, what are dissociation constant? Dissociation is freely reversible. At equilibrium, ratio between dissociated and undissociated particle is a constant. This constant is called dissociation constant, K. And give formula like K is equal to hydrogen concentration into anion concentration divided by HA. There is molecule. Understand? This is dissociation constant. Next, acidity of solution and pH. Acidity means acidity of solution measured by noting the hydrogen ion concentration in the solution and obtained by the equation HA K into acid by base or HA divided by A. Okay, where K is the dissociation constant, H is the concentration of hydrogen ion, A is the concentration of anion or conjugate base, HA is the concentration of undissociated molecule. Acidity of solution is measured by noting the hydrogen concentration in a solution. This is hydrogen, this is constant, acid by base, where K is the dissociation constant and hydrogen is the hydrogen concentration and is the anion or conjugate base. Okay. Make it easier, solution express H plus concentration is the negative of logarithm, where base 10 and hydrogen concentration and design as pH. pH is negative logarithm of hydrogen ion concentration. Okay. pH value is inversely proportional to acidity. Lower the value means higher the acidity. Lower means higher acidity, higher means lower acidity or towards alkalinity. Okay, so this is very basic thing. All have to be familiar with class 12 or HS. This is very important. Henderson and Hasselbalch equation. Okay. This denotes pH is equal to pKa plus log of base by acid. Where pH is equal to pKa plus log salt by acid. pK means where pH is where hydrogen half dissociates, acid is half dissociates, pK. Everyone should know this formula, Henderson and Hasselbalch equation. This is one of the most important topic. Next, so with this formula we can find out what is the pH, normal pH of our body. Example, say in case of bicarbonate buffer, which is in metabolic regulation, the pK value of carbonic acid is 6.1. Normal plasma value bicarbonate 24 millimole per liter. Carbonic acid, which is respiratory regulation, is 1.2 millimole per liter. If we plot this in this formula, we can find that pH is equal to pK, that means 6.1, first log of base, that is bicarbonate, by 24, divided by carbonic acid 1.2. By this way you can find out 7.4 with this formula. Formula is called Henderson-Hasselbalch equation. Okay, next is buffer. You may ask in the exam short note about buffer. So everyone should familiar with this term buffer, know this buffer also. He may ask in exam about short note, 5 marks short note, buffer. So you have to mention these points. Buffers are solution that can resist change in pH after addition of acid or alkali. There are two types of buffer, where mixture of weak acids and their salt with a strong base, or mixture of weak base, their salt with a strong acid. Okay. Few example like bicarbonate buffer, acidated buffer, phosphate buffer, etc. So factor effect, pH of a buffer. pH buffer solution determined by two factors: value of pK. The lower the value of pK, lower is the pH. Best pK value should be around 7.4. Okay. The ratio of salt to acid concentration. Actual concentration of salt and acid in the buffer solution may be varying widely with no change in pH, so long as to ratio of concentration remains the same. Say example, if you given a buffer, you start to add in some acid or alkali. Say I given 1 liter of buffer and then you add strong acid like hydrochloric acid. You have to require 2.5 of hydrochloric acid. That means buffering capacity is 2.5 of the solution with hydrochloric acid. That is called buffering capacity of a solution. Understand? These are basic things. So buffering capacity determined by actual concentration of salt and acid present as well as by the ratio. Buffering capacity is the number of gram equivalent of strong acid or alkali, which is bring about change in pH of 1 unit. I forgot to mention 1 unit. Say if you given a solution of 7, pH is 7, so you have to change it up to 8 after addition of strong alkali like sodium hydroxide. Then the buffer is called, say example I given 2 gram of sodium hydroxide, so buffering capacity is 2 of the solution. By this way. Okay. So the buffering capacity of buffer is defined as the ability of the buffer to resist changes in pH when acid or alkali is added. That is called buffering capacity. Buffer, how do buffer act? Buffer solution consists of mixture of weak acid and base and its salt. To take an example, when hydrochloric acid is added in acidated buffer, the salt react with the acid forming the weak acid, acetic acid and its salt. Similarly, when the base is added, the acid reacts with it forming salt and water. Thus changes in pH are minimized. Like acetic acid with addition of strong alkali give rise to sodium acetate plus H2. When give HCl with sodium acetate, it give rise to acetic acid plus NaCl. By this way, buffer act. Clinical application of Henderson-Hasselbalch equation: the pH of buffer on addition of known quantity of acid or alkali can therefore be predicted by the equation. Moreover, the concentration of salt and acid can be found out by measuring the pH. The Henderson-Hasselbalch equation therefore has greater practical application in clinical practice in assessing the acid-base status and predicting the limit of the composition of body buffer. Next, effective range buffer. pH is when pH is equal to pK, the buffer is most effective. The effective range of buffer is 1 pH unit higher or lower. Say example, a given buffer is pH 7.0. So buffer capacity should be less than 8 and more than 6. Understand? You understand, no? Say example, I given a buffer which pH Example 7.0. So buffering capacity best around 6.0 to 8.0 in this range. And in our body, best buffering capacity should be around 7.4. The closer to 7.4, the best buffering capacity. Next come how our body maintain pH. In our body, normal pH range 7.35 to 7.45. Less than 7.35 means acidity, more than 7.45 means alkalinity, acidosis or alkalosis. Less than 7.35 means acidosis, and more than 7.35 means and this pH range maintained by buffering system in our body. Okay, so the first defense of this buffer system maintained by blood. Normal blood pH. Anyone? What is the normal blood pH? 7. This is range. If I say suddenly what is the normal range of what is the normal pH of our blood, you tell 7.4. Okay, and this is range. 7.5 to 7.4, there is range. Okay, so in our body this pH range fairly maintained by our system. First system is blood. Okay, this is called first defense system of in our body. Second system is respiratory system, and third defense system is renal mechanism. These three systems work in our body to maintain normal pH range. If the pH range goes very down, say less than 7, people will die. Not possible to die, not possible to live as normal human being. Is more than 9, people will die due to alkalosis. And you can't believe, in ICU setup, majority of people die because of the derangement of pH. Because the patient admitted in ICU, they often have multi-organ failure like kidney failure, lungs failure, heart failure. Okay, so their body cannot maintain this normal pH. So maintenance is very, very important in order to survival. So in our body, we have very good buffering system. In extracellular fluid, there are 3 types of buffering system. This is called bicarbonate system, phosphate system, and protein buffer system. In extracellular fluid. In intracellular fluid, there is potassium, hydrogen phosphate, and potassium phosphate. There is phosphate buffer is there. The protein buffer with potassium is there, and bicarbonate buffer is there. And in RBC, hemoglobin buffer is there, phosphate is there, and bicarbonate buffer is there. So you may ask in a Viva exam, tell me what is the buffering system in extracellular fluid. You have to answer this. Like bicarbonate buffer, as blood is in extracellular space fluid, so this thing also present in blood. Bicarbonate, phosphate, and protein buffer. Okay, let's discuss one by one. Blood buffer. There are 3 important blood buffer, as I told, to regulate pH. Bicarbonate buffer system, that is bicarbonate by carbonic acid, the ratio of 20:1, and pK of carbonic acid is 6.1. Understand? Phosphate buffer, here disodium hydrogen phosphate with the ratio of this one, disodium hydrogen phosphate by sodium hydrogen phosphate, ratio is 4:1, pK value 6.8 of this one, sodium hydrogen phosphate is there. This is phosphate buffer. And protein buffer is albumin, hemoglobin, because they have imidazole group and pK value 6.1. There is around 7.4, near to 7.4. So work very well. Understood? So in our buffer, there is 3 types of buffer: bicarbonate buffer, phosphate buffer, and number 3 is protein buffer. Bicarbonate buffer system means it accounts 65% of buffering capacity in plasma and 40% buffering system in whole body. Bicarbonate buffer system regulated by kidney, which is metabolic component, and carbonic acid is regulated by lungs, respiratory system. It is called respiratory component. Normal bicarbonate, as I told earlier, 22 to 26 millimole per liter. Normal carbonic acid concentration blood is 1.2 millimole per liter. The pK value 6.1. Next is, if you substitute this pH with the equation, Henderson-Hasselbalch equation, pK value of carbonic acid 6.1, bicarbonate 24, carbonic acid 1.2, then we found 7.4. As I told earlier. Why bicarbonate system is more important? Because presence of bicarbonate in relative high concentration. It is 20 times higher than carbonic acid, and it denotes alkali reserve in our body. Partial pressure of carbon dioxide and oxygen are expressed in millimeter of mercury in conventional system, but international unit expression kilopascal is used. 1 kilopascal means 7.4 millimeter mercury. Now arterial PCO2 5.3 kilopascal and arterial PCO2 13.4 kilopascal. Alkali reserve, bicarbonate represents the alkali reserve, and you have to present in sufficient amount. If alkali reserve goes down, what happens? Body cannot utilize ion, hydrogen ion. As a result, patient develop acidosis. Eventually, he may die. Anyone tell me what is the function of, what is the regulatory thing which for normal enzymatic activity? Anyone remember? Normal enzymatic activity also depend on pH, temperature, concentration, like this. Okay, so pH fluctuate very high, enzyme activity will be gone, cellular function will be gone. Okay, this is why I am stretching. pH is so important. Next is phosphate buffer. In our blood, second buffering system is phosphate buffer. It is mainly in intracellular buffer, but also present in extracellular space. And pK value is 6.8, phosphate buffer. Next is hemoglobin buffer system. Hemoglobin system is handle the major load of volatile acid like carbonic acid, carbon dioxide, volatile gas. Plasma protein have 1/3 of buffering capacity of hemoglobin when compared by weight. Hemoglobin accounts for 6 times the capacity of buffer hydrogen concentration when compared to plasma protein buffer capacity of non-bicarbonate buffer is 63 millimole per liter, P 7.2. When hemoglobin is oxygenated hydrogen ion are released in lung, and on deoxygenation hemoglobin bind hydrogen ion in tissue. I am coming later, so you remember this. So three systems is that there: bicarbonate system, in blood 3 type system. This is the first defense system, blood. bicarbonate buffering system, phosphate buffering system, and protein buffer system. This is alkali reserve. Next, respiratory system. Respiratory system, how does it work? By hypoventilation or hyperventilation. What is the normal respiratory rate of an adult person? Normal 12 to 16 to 18 or 20, less than 20, more than 12. Okay. In case of neonate, newborn child, it is high, more than 30 or 40. Less than 40, more than 30. Okay. So any adult person, say respiratory rate is 30, that is hyperventilation. Less than 10 means hypoventilation. So we will be read in physiological lecture classes what is the reason behind hyperventilation or hypoventilation. Okay. In acidosis, there is hyperventilation. Alkalosis, hypoventilation. Acidosis means to blow out more carbon dioxide. Okay. So how does it work? In acidosis, where pH is less than 3.5, lung blows out carbon dioxide. Carbon dioxide dissolve in tissue and form this one carbonic acid. How it forms? Carbon dioxide react with water with the help of enzyme carbonic anhydrase. It forms carbonic acid. This carbonic acid can dissociate and form hydrogen ion and bicarbonate ion. Okay. In acidosis, more carbon dioxide is gone. As a result, this level is fall. This level concentration come down. As a result, there is less carbonic acid formed. That means less hydrogen ion forms. By this way, acidity is corrected by hyperventilation. Okay. In alkalosis, where pH is more than 7.45, lungs retain carbon dioxide by decreasing the respiratory rate. Okay. So there is retention of carbon dioxide means there is more carbon dioxide react with water and form carbonic acid. This carbonic acid dissociate and forms bicarbonate and hydrogen ion. By this way, pH decreases. Okay. In hemoglobin, in tissue level, where carbon dioxide is generated, it combines with water and it converts into bicarbonate. Okay. And by enzyme carbonic anhydrase, this carbonic anhydrase generate hydrogen ion. This hydrogen ion react with hemoglobin and form reduced hemoglobin. Then this hemoglobin with circulation reaches in lung where their oxygen level is very high. So in lungs, reduced hemoglobin react with water, oxygen and forms oxyhemoglobin and hydrogen ion release. This hydrogen ion reacts with bicarbonate, forms carbonic acid. Then carbonic acid broken down with the help of enzyme carbonic anhydrase and form water and carbon dioxide. Okay. And then carbon dioxide blows out. By this way, hemoglobin help in respiratory system. Everyone understand these facts? Being a doctor, as a medical student, these facts should be on your lips. Acidosis, alkalosis, metabolic alkalosis, you read throughout your MBBS course. The first defense system is blood by some buffering agents in present in blood, bicarbonate buffer system, phosphate buffer. Are you feeling sleepy? Are you feeling sleepy? You are drowsy, no? Bicarbonate system, phosphate system and protein buffering system. This is first line of defense. Second line defense by lungs. How does they work? This is a mechanism by hyperventilation or hypoventilation. When there is acidosis, lungs tries to blow out more amount of carbon dioxide. When there is alkalosis, lungs want to retain carbon dioxide. Next is hemoglobin. Hemoglobin work in peripheral tissue where their carbon dioxide generate. The carbon dioxide form carbonic acid. Carbon dioxide dissociate and form hydrogen ion and bicarbonate. Hydrogen ion react with hemoglobin, forms reduced hemoglobin. This hemoglobin reaches in lung where oxygen level is very high and it forms oxygenated hemoglobin and is hydrogen ion concentration. Hydrogen ion is released. This hydrogen ion react with bicarbonate and form carbonic acid. Carbonic acid then broken down into water and carbon dioxide blows out via expiration. Okay, this is second lemma difference. Next is renal mechanism. This is one of the most important mechanism. You may ask in the exam. Please write down the renal mechanism of acid pH maintenance. So everyone should be familiar with this mechanism. There is 4 types of systemic work with renal mechanism. First one is excretion of hydrogen ion. Next one is reabsorption of bicarbonate ion. I am telling renal mechanism is one of the most important topic. There is 4 ways of renal mechanism works. First one is excretion of hydrogen ion. Second one is reabsorption of bicarbonate. Third one is excretion of titratable acids, and fourth one is excretion of ammonia. Okay. So you may ask in the exam, write down, you may part of a long question or maybe a short note. Renal mechanism of pH maintenance. Okay. So how this work? Just come with the graph. Say this is in proximal communicable tubule of glomerulus in kidney, where this H1 is form broken down of carbonic acid. This carbonic acid formed because of reaction of carbon dioxide and water with the help of enzyme carbonic anhydrase. This carbonic acid dissociate into hydrogen ion and bicarbonate. Bicarbonate readily reabsorb in the plasma and go in alkali reserve. And this hydrogen ion is excreted in the tubular lumen, and this hydrogen ion is excreted via urine. Understand? Along with sodium ion, it reabsorbed in proximal communicable tube and again it reabsorbed in plasma. This is the first mechanism how hydrogen ion is excreted. So there is excretion of hydrogen ion means acidity lowers and alkali also dissolved. Yeah. This is the first mechanism. If you understand this graph, then it is very easy to understand. Okay. I am taking from Vasudevan, you can read from Vasudevan also. Next is reabsorption of bicarbonate. Here bicarbonate is formed again in the tubular cell, and this bicarbonate is reabsorbed by tubular cell and hydrogen ion is excreted. This hydrogen ion, where in filtered out sodium bicarbonate, is broken down into sodium ion and bicarbonate. This hydrogen ion react with bicarbonate form carbonic acid. With the help of enzyme carbonic anhydrase, it broken down into carbon dioxide and water. Sorry. This carbon dioxide is dissociated back in the tubular cell. Okay, and react with water from bicarbonate, carbonic acid, and again from bicarbonate and directly reabsorb. By this way, reabsorption of bicarbonate occurs. Okay, in tubular cell. This is the second mechanism. Next is excretion of titratable acid. In tubular lumen, disodium hydrogen phosphate is excreted out, come out. By it broken down into sodium ion and sodium hydrogen phosphate. It reacts with excreted hydrogen ion and forms sodium dihydrogen phosphate, and it is excreted. By this way, excretion of titratable acids has occurred. Okay. Along with this acid, hydrogen ion also excreted. Understand? This is the third fact. And next one is excretion of amide. Here, this ammonia is formed from glutamine by the enzyme glutaminase with addition of water, and it forms glutamic acid. This ammonia is dissociated out in tubular lumen in collecting duct, okay, and ammonia is react with hydrogen ion and forms amide ion, and this is by this way hydrogen is trapped and excreted. Okay, so by this way hydrogen ion along with ammonia is excreted in tubular lumen. So these are the four mechanisms by which pH is maintained in our body. So this is our first class. Next class we will going to discuss the disorder about pH in our body. Okay. So today just see here bicarbonate when it is less than 22, that means called metabolic acidosis. And where it is more than 26, it is called metabolic alkalosis. Okay. When PCO2, there is partial pressure of carbon dioxide, normal range is 35 to 45 millimeter of mercury. When PCO2 is less than 35, that is called respiratory alkalosis, and when PCO2 more than 45, that is called respiratory acidosis. Okay, so when you going to diagnose patient, we say you have come with ABG report. How can you diagnose that? First of all, you see what is the normal pH, what is the pH you found in the report. Say pH is less than 7.35, that is called acidosis. Then next step is what kind of acidosis? Is it metabolic acidosis or respiratory acidosis? Then you have to see the bicarbonate buffer. Say bicarbonate buffer is less than 22, that is called metabolic acidosis. Or when there is PCO2 is more than 45, that is called respiratory acidosis. You have to see which one is correspond with the pH. Okay. Next one, you have to see the PCO2 level and with the compensation. By the way, I will discuss in next class.